Okay, today we're going to be taking a look at genetics, and this corresponds to chapter 11 in your biology textbooks. And basically, when we're studying genetics, we're looking at the study of heredity, or in other words, the passing on of characteristics from one organism to another. This goes from everything that makes you, you. So let's go ahead and take a look at that by starting off way back at the beginning with Gregor Mendel. Okay, he was an Austrian monk who lived from 1822 to 1884. And he used a scientific approach to study inheritance. He used like the scientific method. All right, he was able to sit down and approach genetics in a way that nobody else had done before. All right, he's considered the father of genetics, and of course there was no knowledge even in his time of DNA. So he was going purely on the scientific process. And of course he studied inheritance patterns in pea plants. So why would he pick pea plants of all things? Well, first off, they have great contrasting traits or characteristics. So in other words, things that you could easily see without really any major scientific equipment. They reproduce sexually, and they do it very quickly, and of course have many offspring. So once again, this gives you a large sample size. All right, they're very accessible, easy to handle. So it was quite easy for Gregor Mendel to use the pea plants in order to study this. Now, he used a process called controlled reproduction. If we take a look at the diagram of the flower we have over here, I want you to pay special close attention to the stigma that you have here and the stamen that you have over here. All right, the stamen, all right, produces pollen. And of course, that's the pollen that sometimes you wake up when there's a high pollen count, you can see it covering various things around your yard or your house. All right, and the carpel or pistil here, which includes the stigma, the style, and the ovary, produces the eggs. All right, now, what Gregor Mendel did is he removed the immature stamen, and of course, this prevents self-fertilization. The stamen will produce the pollen, and of course, that will make its way down in the stigma, and of course, cause the reproduction to take place. All right. So instead of using letting the plant do it itself, he would cut off the immature stamen, as you see here, and then he would use kind of like an artist paintbrush to control the reproduction process and painting it on the stigma of the plant, the pollen that is, so the conception could take place. Now, sexual reproduction in plants, fertilization is going to occur when a male and female sex cells join together, just like in most cases any other type of sexual reproduction. And of course, cross-pollination here is pollen from one plant fertilizes or pollinates the eggs of another plant. And this is what Gregor Mendel was really going for. He was going to start crossing plants and find out what he would get in the offspring. All right. And of course, self-pollination is when the pollen and eggs come from the same flower. All right. So he wanted to see, okay, well, if I cross one flower with another, what am I going to end up with? And of course, Mendel looked at 7 traits of pea plants, and you can see the trait and the variance of each trait below. We have the height, either tall or dwarf, flower color is purple or white, and flower position, axial or terminal. All right, so in other words, it'll branch off or it terminates. A seed color, whether it was yellow and green, the shape of the seed, round or wrinkled, the pod color, green or yellow, and whether the pod was smooth or constricted so that you could actually see the seeds within it. So those were the 7 traits that Mendel looked at. And of course, let's take a look at Mendel's experiment. Mendel identified stocks of plants that were true breeding for a particular trait. Tall plants, obviously self-pollination, and of course, you ended up with all tall offspring. All right. Mendel decided to cross 2 true breeding plants with contrasting characteristics. So he wanted to take 2 plants that he knew were going to always, through self-pollination, produce the same trait, and he wanted to cross-pollinate them. So, for example, he took purple flowers and white flowers because he wanted to see what would happen. All right. He called them the P generation, for parental. So that's where we get the P from. All right. So what do you think the offspring look like here? We got purple flowers and white flowers. Now, a lot of you might go, all right, well, it's probably a cross somewhere between a purplish white. All right. Well, let's take a look. So Mendel, of course, removed the stamens from the purple flower, and he transferred the pollen with a paintbrush to the carpel of the white flower. And, of course, the pollinated carpel matured into a pod, and we end up with the first generation that you see. So we got this first generation, and as you can see, all the flowers were purple, even though he crossed a true breeding purple with white. All right. So F1 generation is that first generation offspring that we see at the bottom that are all purple from the cross from the P generation, which is the parent. All right. Now that F1 comes from filia, or sun, for which is sun in Latin. All right. The offspring were 100% purple. So Mendel's going to repeat this experiment over and over again, and he's going to observe similar results with all 7 traits that he ended up studying. He called this F1 generation hybrids, all right, because the parents obviously had 2 different traits. Even though they were expressing one, he was curious to see what would happen after he took the F1 generation and crossed them. So let's take a look at that with our second generation. Mendel let the F1 generation self-pollinate. In other words, didn't cut off the stamens. All right, they just pollinated and did their own thing to create an F2 generation. And sure enough, the white flower that originally was lost in the F1 generation reappeared in the second generation. All right, and it appeared in a ratio of 3 purple to 1 white. Now, it's not like every time the plant was going to have 4 flowers, 3 of them would be purple and 1 would be white. Well, that was the ratio overall after many experimentations that he ended up coming up with. It's just a probability of saying every time that that flower would produce another flower in the F2, there was a 3 in 4 chance that it would be purple and a 1 in 4 chance that it would be white. All right, here we have a diagram showing our P, our parental generation, and of course our F1, which are our hybrids. And of course, through self-fertilization, we end up with that 3 to 1 ratio in F2. So Mendel formed some principles on this, and of course, in modern terms, if we take a look at them, he says that traits are passed from one organism to another by units called genes. All right, and there are different forms of genes called alleles. Now, alleles is an important term because a lot of people get it confused with a lot of other terms. But make sure you pay attention to what it's saying. There are different forms of genes called alleles. All right. Example would be height. The gene is height, and the allele could be tall or dwarf. So genes is the actual trait, height, and there can be different forms of that gene, tall and dwarf. Those are the alleles. Mendel also stated that genes are inherited in pairs, 1 allele from each parent. All right. If you get the same gene from each parent, identical alleles, tall and tall, let's say here, that would be homozygous. All right. If you got a tall from one parent and a dwarf from another, all right, that would be heterozygous. That's where we have 2 different alleles. Okay. And what do you think here? For tall or dwarf, resorting back to our first example here, which one would show? That's right, it would be the tall, because we know that to be the dominant one. In a hybrid, only one dominant allele, T, will be expressed or seen. The other allele is going to be recessive. And of course, we're using T's here. A lot of people use R's. It really depends on what you're using. You can abbreviate it really any way you want as long as you know and can tell people with a key what it is. Also, alleles segregate or separate from each other during gamete formation and rejoin during fertilization, obviously to make the offspring. Okay, so remember from meiosis where we had that separation of alleles, and they're segregated out, and of course they rejoin during fertilization, one from the male and the female in sexual reproduction, to produce the offspring. So let's look at the principles of this segregation. We know that it happens in meiosis, where we go from a diploid parent to a haploid, all right, where we only have one set of information. And you can see that in the F1 there. Now, if we look at the first one, they are both hybrids. They have a dominant and a recessive. We would call them heterozygous. Now, that segregation is going to take place of those alleles, big Y, little y, big Y, little y, as you can see right here in the diagram. And of course, we can see the combinations that we can come up with. Homozygous dominant, heterozygous, heterozygous, and homozygous recessive. Now, if we relate that back to our purple and white flowers, we would see if dominant's purple, which it is, purple here. Now, there's a purple and a white, but the purple will be expressed, and here is our white will be expressed. That gives us our 3-to-1 ratio that we saw earlier. And of course, what process segregates the alleles during gamete formation? Well, you guessed it, meiosis. All right, there's our diploid parent. Then we have interphase, and then we go through the first set of meiosis, and then we go through our second division of meiosis, and of course that's where we have that segregation take place, and we have the different alleles in each gamete. Alright, that's where we're going to end today with this section here, so make sure you take a look over this, stop and repeat things as many times as you need to. Take some notes on this, and if you got questions, write them down. That way you can talk to me during class, because I'm sure somebody else has the same question as you, and we can go over it again. All right, thanks guys. See you soon.